more information. So our 1,2 is more of a kinetic result. Okay, so it happens very, very fast. So what we're looking at with our kinetic result is trying to neutralize something wicked fast because it's wicked unstable. So in the case of your 1,2 additions, what we're saying is your nucleophile is wicked negative. Apparently I like the word wicked today. Wicked negative. Okay? So very, very, very unstable. That's what our nucleophile is in the 1,2 addition. And in all the cases we've looked at, we've pretty much just said nucleophile negative, carbonyl positive, it does the deal, you don't worry about it. What happened when we moved into this next section, though, is we looked at a pseudo-new functional group, our alpha-beta unsaturated ketones, or carbonyls, I should say. And in the case of our alpha-beta unsaturated carbonyls, we don't have just the electrophilic carbonyl. We also have an electrophilic carbon at our beta position of our alpha-beta unsaturated carbonyl. Okay, so there's 2 reactive sites. With there now being 2 reactive sites, we have to decide which one is the one that reacts. And in most circumstances, we could usually come up with some explanation. Well, this one's more positive, so this one works. We're still in that carbonyl chapter, so our carbonyl chemistry kind of goes a little bit haywire. And what we're looking at is not so much this one's more positive, so it reacts, but this one generates a resonance stabilized product or a resonance stabilized intermediate. Something about that reaction, reacting at that site, generates something that is more stable than where we were previously. Okay? That's kind of the intent on it. And we have seen this already. When we looked at our conjugated dienes, we had multiple types of additions across our dienes. It wasn't just a single double bond. Should I draw up an example of that?
Yes.
Because I really am just kind of spitballing right now.
So before the deal's over, we're talking.
It was the same kind of idea. I think it was in that same chapter. Okay, if we have our diene and we reacted this with HBr, we had a bunch of possible products that could have come out of this. Right? We could have generated, sorry, I have to look up at what I'm drawing because it's in an odd spot for me to see, and I drew it wrong. Shit. Let me try that again. So start up. We could have ended up with a product where we had H and Br, and our double bond did not move. Or we could have gone through and gotten a different product where we have our H and our Br. Right? You guys remember that section? So what's happening is we have a resonance stabilized structure. And in this case, both those products are pretty much equally favored. Okay? Because we look at our alkene, and the alkene, which is our highest energy species, in both cases, is di-substituted. There's no difference between those. But if we simplified this and erased a carbon, now which product is favored? Top or bottom? Okay, so we got competing clues or competing information. Good. So let's fight about it. Okay. Why do you say the top one? I forgot. Did you say top?
I did.
Okay. Why did you say the top one?
Because the carbocation, or the carbon that it goes to, when you have a charge on there, it's more stable.
So what you're saying is the top one is favored. Let me add a different color in there. I may not be able to spit it. Top one is favored because stable intermediate.
Yeah, more stable than the
What about the bottom one?
It's going to stabilize the unstable, I thought.
Okay, so you said the bottom. Why the bottom one?
Because won't it stabilize when you have that, the double bond is stabilizing it because it's
If we look at the product itself, the double bond being our highest energy thing in our products. Okay? The pi bond is our highest energy structure. The pi bond is at a lower energy because it's a di-substituted product. So di-substituted double bond or di-substituted alkene. Okay? So that's an interesting dilemma. But realize we're talking about two different things. Teresa, you were talking about the intermediate.
Yeah,
Megan, you're talking about
the product.
product. Okay? Well, which one dominates?
It depends if it's SN1 or SN2.
No, it's not substitution.
I mean,
Elimination.
It's not an elimination. It's an
Oh,
addition.
addition.
It depends on the reagent conditions that we're running under and temperature conditions. What we've got in this case for that top one is the kinetic product, because the intermediate was the most stable, which means the transition state is lower, we get a faster product. If we move to the bottom one,
That's thermodynamic.
we have a thermodynamic product, because the alkene, our final product, is at a lower energy. Okay, so we get a kinetic versus thermodynamic. And the way we would favor each of those is by changing our reaction conditions. We can't change what we're adding to it because it's H and Br, and we're getting these two products. But what we can change is the temperature. What happens when we drop the temperature?
When it's colder.
It's cooler, it's kinetic.
It's going to favor the kinetic because we now haven't given our reaction enough energy. If we look at an energy diagram, we go at our reactants to our products versus reactants to our products.
For thermodynamic.
Our kinetic one,
Can't overcome that.
with such a low energy, with low temperature, can at least clear that activation barrier because it's lower. And we would expect to see the kinetic product. Okay? But if I want the thermodynamic product, what do I want to do?
Increase the temperature.
Jump the temperature upwards. So if I provide enough energy to now clear that high barrier, okay, yes, I will still get the kinetic product.
Not
But
too
I've
much of it.
also provided enough energy for the kinetic product to go in the reverse direction. So the kinetic product is now in an equilibrium situation. Okay? But the thermodynamic product is not. Okay? So the thermodynamic is now only favored one direction. So if I provide enough energy, yes, I clear the activation barrier for both. So both products are possible, but only one can do the reverse reaction. All right, so if I add a lot of energy, I get the thermodynamic product because the kinetic reverses its direction and I lose the kinetic product. Kind of makes sense? When we go through and look at, I'm going to lose all my information. Oh well. What do I do? Our conjugate addition mechanism, we have the same situation. Our 1,2 addition is our kinetic product. Okay? We're taking our most positive and our most negative, we combine them immediately, stabilize, get rid of those reactive intermediates as fast as possible. If we go through and look at our 1,4, that's really our thermodynamic. Okay? So what we're looking at is how are these different things translated out into what we end up seeing in our end results. Okay? That's kind of how we're evaluating them. So it's very similar to our idea of what we've seen before. So when we look at our 1,2 additions, we're trying to neutralize our charges as fast as possible. So we're just trying to do the fastest reaction, neutralize those charges right away. In our 1,4 addition, our nucleophile is still present, but it's not as strong as a nucleophile. Say that right? It sounded kind of weird. Right?
Say that again.
What,
It's-
in our 1,4?
In our 1,4, our nucleophile is not as strong of a nucleophile as it is in our 1,2.
Correct.
So-
It's the heat that you're adding to that. It's a weaker nucleophile, but you've added heat. Correct?
We didn't even look at temperature. It's just a weaker nucleophile. So under that weaker nucleophile circumstance, we can now spend the time to find the most stable product possible. because I don't need to neutralize the negative or the positive right away.
You have time, is what you're
We've
saying.
effectively increased the
Created time.
created time to allow for our reaction to find this is the best product possible, okay, because it's the most stable. Okay? Within that, this is where you had problems with Janine. Our nucleophile, we need to be resonance stabilized. It needs to be stabilized somehow. So our most common nucleophile for the 1,4 addition is what's known as our Gilman reagent. Okay? And that's our copper reagent. So it's copper with some R group, okay? Some carbons. And that has to be generated with our lithium. So that Gilman reagent is our classic one. Okay? So that is our negatively charged carbon. That is going to be our nucleophile, our classic nucleophile for the 1,4 addition. Yes?
I thought like the 1,4 addition is favored when there is reversibility. So how can it be the thermodynamic when?
It's reversibility in our kinetic step. So our kinetic step is reversible, which allows for the thermodynamic step to occur. So if we looked at our energy,
You get both products,
if we get both products will occur,
but the kinetic reverses.
but our kinetic will reverse. Okay? If we can't reverse,
Right?
if we cannot reverse,
The kinetic is always going to
then we're always going to get the kinetic. We don't have a choice because there's no way to reverse the reaction. Okay? And that's why if we have a nucleophile that is resonance stabilized, we get the reverse. But if it's not resonance stabilized, we can't do the reverse because our difference now in energy between our reactants and products is so high that I can't get the reverse reaction to occur. So even if it has a low activation barrier, it must be able to do the reverse. We have to have that energy threshold. And what we're saying is in the 1,2 additions with our Grignard reagents, because that's our classic example, Grignard, for our 1,2, and in our other one, we've got our Gilman reagents, G and G, that's nice, isn't it? Our Gilman reagents are stabilized. We've stabilized that negative, so it does allow for a reverse reaction of our 1,2 addition. That reverse then gives us the opportunity to do the 1,4. The 1,4 won't reverse because it's thermodynamically more stable. Okay? Bottom line, what you need to be able to recognize is the difference between a Grignard reagent and a Gilman reagent and how that chemistry changes. If we have a Gilman reagent, we get the 1,4 addition. If we have a Grignard reagent, we get the 1,2. Okay? The reason why you don't necessarily need to know, okay, particularly with 3 weeks left in the semester, okay, what you should be focusing on is that under the 1,2 addition conditions, you have a negative nucleophile that is not stabilized. So you're looking at sp3 carbons. Okay? If we have a nucleophile that is stabilized, you're looking at a Gilman reagent, you get the 1,4 addition. Okay? What makes it stabilized? Well, our classic example for our Gilman is that alkyl cuprates. Okay? So that alkyl group attached to the copper is now a nucleophile, but the copper stabilizes it enough that we see the 1,4 addition. Okay? Other examples. Cyanide. Okay? Why does cyanide act as a Gilman reagent?
Because it's carbon nucleophile.
They're both carbon nucleophiles. Even the Grignard gets you a carbon
Well,
nucleophile.
that's true. Is it because of the nitrogen? Stabilization. with-
There's a stabilization where we could take those electrons and push them onto our nitrogen. Okay, so there's a pseudo-resonance stabilization. It looks a little bit weird, but we could do that. Okay. There is also a hybridization stabilization. That carbon is sp, which means it's more electronegative. It's now stabilized that negative charge a little bit better. Okay. Just for giggles,
It's SP hybridized.
it's sp hybridized, yeah.
No, it's not a straight up SP.
I'm actually not positive on this, but I'm hoping I'm right.
Let's try it.
Methoxide. Where would we expect that reagent to attack? Okay, why 1, 2? Okay, we'll walk through my explanation too. Why do you think 1, 2?
Because it's not very, to stabilize.
There's no resonant stabilization, right? But the argument that I would make is that we said our carbon from cyanide was a Gilman reagent, giving us 1, 4. Because that carbon is sp hybridized, making it more electronegative. So if we put a hydrogen on there, we'd say that that carbon is more acidic, and it would ditch off that hydrogen pretty quickly. What's the relative acidity of an sp carbon versus an OH? So you'd have to know something about your pKa's. The pKa of acetylene, 25 to 30. pKa of an alcohol, 16. Okay. So we're right, there is no resonant stabilization, but there is another form of stabilization in methoxide. What is that stabilization? No. Oxygen is more electronegative than carbon. The sheer fact that oxygen is more electronegative is enough to stabilize that negative charge. So I would argue you're going to get a larger concentration of the 1,4 addition. How do we know? We'd have to look it up, see if somebody's done it. I'm sure somebody has. Okay. Another reason why we would get the 1,4, what happens if we did the 1,2? What product would we get? Can't kick anything out.
No, but if it attacks the carbonyl, you already have
What product do you get if you have the methoxide attack a carbonyl? It's a particular functional group. You get a hemiacetal. What do hemiacetals do?
They're not stable.
They're not stable, and it reverts back to our carbonyl. So even if the 1,2 was a possibility, it generates a very unstable intermediate that very quickly reverts right back the other direction. So 1,2 can't happen. 1,4 is all that's left. Okay? So, back to what's happening with all of this enolate stuff. So one of the reasons our 1,4 is our favored product is that the intermediate that is produced in our very first step, okay, under both conditions, in the 1,2 addition, our very first intermediate is not resonance stabilized. Okay, there is no resonance for that. We have generated a negative charge that can't go anywhere. It's stuck. What happens in the 1,4 addition?
What was the question?
What we're looking at is the intermediate of our very first step in both the 1,2 and the 1,4. In the 1,4, what do we end up doing with that negative charge? We have resonance stabilization. Okay. That resonance stabilization, because this was an alpha-beta unsaturated carbonyl to start with, happens to be an enolate. That's it. That's the only reason we're calling it an enolate is because because we started with an alpha-beta unsaturated ketone. Our product, if we attack in the 1,4 type addition, is an enolate. That enolate is resonance stabilized. Okay? So that's where the term enolate is happening within our mechanism. It's solely that because we started with the alpha-beta unsaturated carbonyl, we have to generate an enolate if it attacks at the beta position. We don't have a choice. That's what's happening. So we could treat the enolate as pretty much the formation of a product. Right? And that is a resonance stabilized product, which now starts to suggest that we could get that 1,4 addition product. Okay? So there's our first occurrence of an enolate. Right? It just happens to be an intermediate within the mechanism. Okay? So where did the second occurrence of an enolate show up? If we now expand beyond this just being a random nucleophile attacking, right, what charge does our nucleophile need to be? Negative. How can I get a negative nucleophile? Give me some options, particularly with reference to what we've just been looking at. Are Grignards negative nucleophiles? Yes. Gilman reagents?
Yes.
Yes. How about the alpha position of a carbonyl structure? Can that be turned into a nucleophile?
Yes.
Why?
Because it has acidic hydrogen.
Kick off the acidic hydrogen, and we have resonance stabilization known as enolates. Okay? So our nucleophile can also be an enolate. There's our second occurrence. Kind of make a little bit more sense. So we are seeing enolates as a term showing up multiple times in our reaction. But by that same token, I start with what functional group? An aldehyde. What do I end with? An aldehyde. Aldehyde's both a reactant and a product. How can that possibly be true? Well, no one seems to challenge that, but when we bring in this term of enolate, now everybody kind of panics a little bit. It's the same idea. An enolate is a particular type of functional group. That enolate can show up multiple times throughout our reaction. Okay? So just because we've seen enolates as nucleophiles and as reactants does not mean that is their only purpose. They can show up as intermediates and they can show up as potentially products as well. Okay? Does that help separate that now, Janine?
Yeah.
A little bit at least? Okay. Ultimately, what you need to evaluate is that when we're looking at the 1,4 addition, your nucleophile is resonance stabilized, or it's one of those special conditions, cyanide or the alkyl cuprates. Okay? Those are our primary focus for 1,4 additions. Okay? So we then looked at our conjugate addition reactions. We've got this Gilman, enolate, and Grignard reagents. We're kind of running that span. Our Grignards are exclusively 1,2. Those Grignard reagents are so negatively charged that they need the biggest positive immediately. So they go extremely fast for that carbonyl carbon, regardless of the structure of any intermediates. What happens with our enolates? Well, enolates are resonance stabilized, and depending on how much resonance, they kind of look like a Grignard, they kind of look like a Gilman. So they kind of float through the middle. So your enolates can favor both the 1,2 and the 1,4. But if I have a doubly resonance stabilized enolate, that now pushes it closer to our Gilman reagent. We expect the 1,4 addition. Okay, our enolates. Within enolates, it doesn't have to be a single enolate. It could be a di-enolate. What do I mean by that?
Yeah, exactly.
Okay, so let's look at that bottom structure. I'm going to cross this part out. Is there an enolate? Can I make an enolate? Yeah. I got hydrogens here. Where else do I have hydrogens? Hydrogens over here. So I can generate an enolate, a negative carbon, at either one of those positions. That is a single enolate, because there's only one resonance option for those electrons. Okay. What do I mean by a di-enolate? Well, in the di-enolate, now let's not ignore the bottom part of that structure. Okay, if we add that part of the structure in, you'll notice that there is yet another position where we can form an enolate. You'll notice I drew it in red. Why did I draw it in red?
It's identical to the one.
It's identical to the other one. And the one in blue, why did I draw that one blue?
It's different.
It's different. What's different about it?
Between the two photons.
Why does that make it different?
It's more acidic.
It can resonate with one. There's our first enolate. Or it could have resonated this direction, which would be our second enolate. That compound is di-enolate resonance stabilized. That is a good Gilman reagent. The red positions would be a standard enolate that floats in between the Grignard and the Gilman. So if those carbons acted as a nucleophile, we'd get a mix of both the 1,2 and the 1,4. Right? With the structure that we've got with our blue nucleophile, because that is di-stabilized, okay, we have two enolate forms. That now pushes it more towards the Gilman reagent. We see exclusively the 1,4 addition. Okay, so when we're talking about a di-resonance effect, what we're saying is that that nucleophilic carbon, as far as an enolate goes, is stabilized either direction, okay, which is what we see when we look at our Michael reactions. If you look at the Michael donors, all of those have what in common? Okay, except for the last two. We'll exclude the last two for the moment. If we take a look at the nucleophile, our negatively charged carbons, what can those negatively charged carbons do? They have how many forms of resonance? Two. They can go both left and right. Di-stabilized nucleophiles. You'll notice the bottom two don't quite fit that pattern. Right? Within those, that's kind of like your sodium borohydride example versus lithium aluminum hydride. There's a threshold where all of a sudden it just changes and we lose that chemistry difference. Right? Your classic Gilman reagent, or what I'm pretty sure is associated strictly with Gilman, is that cuprate, our alkyl cuprate. That is solely going to be our Gilman reagent. That's just like our Grignard, except that copper stabilizes the negative carbon just a little bit better than the magnesium, and that reduces its nucleophilicity enough that we see a 1,4 instead of a 1,2 product.
Because it's more electronegative, right? So it stabilizes it more.
Yes. Sorry. I thought you had it backwards, but you were absolutely right. All right. The other one, why does our nitrate, why is this guy so much better? Why is that one a good Michael reaction? There's only one resonance structure. Finish that statement. Nitrogen's really? Try again. We draw out the Lewis structure. What charge is that nitrogen? It's formally positive. That is a very strong resonance contributor because I'm neutralizing two charges at once. Okay, by shifting those electrons in. Whereas if we go through and look at all of our other atoms, what charge is the carbon in any one of those upper structures? Any of the carbons that I just highlighted in blue, which doesn't show up very well. Let me try that in red. Try, there it is. It is partially positive in all of those cases. So while we can draw resonance there, it's not a massive resonance structure. But if we look at our nitro compound, we get massive resonance because what's the charge on the atom? Formally positive. That's a much larger resonance contributor to stabilize that. Okay?
Because you have
So
a
that
formal positive and a formal negative.
exactly right. Okay? As far as your Michael acceptors, you're looking at the same thing. Putting that alkene, your pi bond, next to some kind of electron withdrawing group. The most common one that you guys will see would be your carbonyls. But it could be a nitro group. It could be a cyanide or the nitriles. All of those have the same ability. Right? Our oxygen is more electronegative than our carbon, so it pulls electron density away so we can get resonance, okay, pushing in. If we go through and look at our nitriles, we still have a resonance contributor, pulling that electron density away, making our carbon even more positive. Right? All of those contributions make those good Michael acceptors. That beta position of our alpha-beta functional group is going to act as our acceptor. Okay, so it will react in this. Why do we call them Michael donors and Michael acceptors? Because this guy Michael was super famous, right? And probably had a lot of money and power and convinced everybody to name reactions after him.
It's just confusing, if you've seen all the lot of structures on the left.
You've seen all of them.
Throughout the yeah, all these
Yep.
different chapters, and like suddenly now it's got a new name.
And now we call them something new.
It's ridiculous.
It is. I completely agree with you. Keep that attitude when you move through and talk to other people about chemistry. Because if we can build up enough of an attitude of, this is crap to have to name something after somebody, maybe we can stop having to bring in these names. It's not going to happen, but it's a nice hope. Hi? So,
can we memorize these, I mean, we have to memorize these 6 to know this?
no, so this is not 6. These are examples. So what you need to be recognizing is your Michael donors, okay, are at least di-resonance stabilized. And we have some special conditions. The special conditions that you should remember would be that copper, the alkyl lithium one. The one with the nitro, I wouldn't worry about. I wouldn't bother to memorize that. If it showed up on a test, what you would do is see, oh, that's a really strong resonance stabilization. If I have that really strong stabilization, that negative isn't as strong of a nucleophile. What am I reacting it with? Am I reacting it with a structure that has 2 electrophilic atoms? Oh, if I do, where is it going to react? Okay, so it's a very special kind of case and circumstance. This is usually why it's reserved for the tail end of second semester organic chemistry. Okay? Questions? Don't shoot the messenger. So, should we work through the mechanism real quickly? So we've got a Michael donor, which was nicely drawn for us with our negative. And we've highlighted the Michael acceptor. That will attack that carbon. But then that carbon has too many bonds. So we have to break a bond. What bond do we decide to break? The pi bond. Why the pi bond? The pi bond is the easiest bond to break. Always break the pi bond first. All right, where do we move those pi electrons? Okay, so you've decided that you want to move it to the neighboring sigma bond. I can accept that. But then how many bonds does that next carbon have? Too many bonds. What do we have to do?
Push it down.
Break another pi bond and put it up onto the oxygen. Right? Does that oxygen has that oxygen exceeded its octet? It does not. So we're fine. We can do that. So we'll continue to add arrows until we've reached a point where we haven't exceeded an octet. All right, and if we draw this out, see if I can attempt to draw this nicely. Was it ET? Oxygen, and I'm going to go through and highlight in red. We got the negative charge there, double bond there. Everybody see it? Okay. Now what happens? Okay, so depending on mechanism choices, we may decide that there's a hydrogen somewhere in solution. So for my arguments, I almost always write HB. Okay? I don't try and associate any charge with that B, because if I put charge on it, I'm now specifying whether it's acidic or basic. Okay? And I don't want to make that statement. So if I've got that, I can say the negative picks up our hydrogen.
What if it doesn't do that?
Give me a second to draw it. Well, actually, keep talking. What do you mean? What if it doesn't do that?
What if you don't have the hydrogen to pick up, so the oxygen pushes back electrons and then...
Okay, give me a sec. I know what you're saying. I will be there. This and this. What if you don't have that hydrogen? What happens?
So then we'll break off the...
We can push those back down, but then what happens?
And then
Too many bonds, so where does that pi bond go? Which bond is easier to break? The pi bond. So where does those pi electrons go?
To that carbon.
To that carbon. What have you just drawn?
A new,
Careful.
it's not the same thing you started with, it's another.
Maybe it'll help if we draw it all out.
Yeah.
Yes, it's an enolate. What's the relationship between my black structure and my blue structure? What was that? They are resonant structures. Okay, so we drew resonance. Ultimately, doesn't matter which one we've got. This is one of the driving forces for our 1,4 addition. We have resonance to stabilize that intermediate. Okay, but now the question is, okay, what do we do? Almost always you'll see these followed by a second condition. What's that second condition going to be? The addition of some form of acid, which could very easily just be water. Does it need to be a strong acid?
No.
No. You've got a negative carbon. Okay? That's a wicked strong base. It's going to pull a hydrogen straight off the water. Okay? So if we then go through and show that, there's my HB again. And I did hear you, Megan. Just give me a second. Drew that off, and we draw out the product of that. Okay, what's the relationship between that lower right and the structure right above it? I'm asking, I'll box it here, what's the relationship between these two structures? It's a big hint.
Enol. Are they towers?
They're tautomers. You have the ketone in one form, you have the enol in the other. They're tautomers, which heavily favors the ketone.
So yeah, why wouldn't it?
Okay. So what is your final product? Your final answer would be that lower right-hand corner. It's the keto form. So it's a question of when do you put on that hydrogen? Ultimately, it doesn't matter when you put it on, as long as you realize that you have to go to the keto form. You do not stop at the enol unless you are explicitly told to stop at the enol, right? Which won't ever happen. Okay? Kind of makes sense? Questions about that?
How would that show up on an exam? Would we want to
What's
be
the
given-
product of these
would
two?
one be- Would we be given like that without the,
What is the most stable?
I mean, it seems like we wouldn't be given,
What you would likely be given, so let's come up with a couple options. So one version of the question is, what is the product? As the most straightforward version. And what they would not give you is that negative charge. They'd say, what is the product of these two structures reacting with each other? Okay, that's probably the most straightforward question. They usually try and mix them up and get a little bit different. The other option would be, let's try it this way. 2, what reactants? And what they would give you is our lower right-hand corner. What were the reactants used to make that? Okay, which means drawing two structures, so they could switch it up. They could say, instead of what reactants, they could say what nucleophile, or source for your nucleophile, okay, or what electrophile was used to make that. I would guess which of the following is the correct electrophile or nucleophile is probably the best way they'd ask it, because what they would provide would be both of those structures and then a couple other ones just to mess with your head. Both of those structures are involved in this reaction, but which one generates the nucleophile? The nitrile structure or cyanide structure. Okay? So that would be a case where you could whittle it down to 2 answers, hopefully pretty quickly, because both of those structures are needed to make the product. But you have to know something about how they react to decide the one on the left is our nucleophile. Okay? Kind of makes sense. I thought I had a third way to ask it, but I don't. Sorry. So that's it. Well, that's almost 4 ways right there. So what are the reactants? What was your nucleophile? What was your electrophile? Okay. Another option could be which of the following is a correct intermediate. And they could show you a whole bunch of different intermediates within it, okay, which could suck. My guess is if they asked you that, they would probably, yeah, actually, I don't know how to do that one. Okay. Other questions? Ta-da, move forward. Enamine, conjugate addition reactions. Okay. We talked about our enamine briefly before. Our enamine was kind of nice because we now have, or when we first formed it, we actually didn't do anything with it. We just said, oh, we've got it. So there's our enamine. Right? If we think about our enamine now under the context of what we just talked about, what can our enamine do? Well, those lone pairs can come down and make that carbon nucleophilic. Okay. You now have a nucleophilic carbon that could potentially go through and do all the same chemistry that our enolates went through and did. But now the next question, of course, is which would we expect it to do? Would we expect it to do a 1,4 or a 1,2?
I would say 1 and 4.
Okay, why 1,4?
You're looking, I mean, when we're talking about that 1, 2, you're talking about a carbon
Yeah.
cases, right?
So yeah, I mean, I'm trying to think of a counterexample. So we could pull sterics. I think sterics would work as an argument.
I'm just saying from the standpoint of you're usually looking at a carbonyl.
But when we
Talking
did
about that.
the chemistry with just a strict enolate, the enolate, we said, could actually form 1, 2, and 1, 4. So just a standard single enolate gives us both a 1, 2 and 1, 4 products.
Okay.
So what would we expect for just the enamine?
The same, essentially.
Okay, we might expect exactly the same thing because you're looking at almost an identical structure. Keyword there being almost. What's the difference between these? Could look at the R group. I'd say there's a bigger difference because the R group comes back to sterics. Nitrogen versus oxygen.
Nitrogen is less electronegative.
Why is that going to help out? Our nitrogen being less electronegative can help put more negative charge or stabilize that negative charge a little bit better so that we form a higher concentration of that. We really do see a 1,4 addition primarily. Okay. So kind of a neat intermediate with that. It also does all sorts of extra things associated with it beyond that, which I don't think you really need to stress about. Okay, so I'm not sure how much more detail I want to go beyond that. Okay. So we end up under other circumstances. So if we looked at our enolates, we expect this is our primary enolate form for a standard enol. If we go through and do the enamine, the one we end up favoring is the kinetic product, not our thermodynamic product. And that has to do with sterics. Those sterics, dang it, I meant to grab a model kit, have to do with the one in our thermodynamic favored or our thermodynamic situation. This carbon, this carbon, this carbon, and the nitrogen are all in the same plane, okay, due to resonance. which also puts this carbon in that same plane. All of those atoms being in the exact same plane, we start to get some steric budding between those atoms, which disfavors thermodynamic formation. Whereas if we go through and look at our kinetic product, we get a similar situation, except now it's the hydrogen, carbon, carbon, nitrogen, and carbon. So when we have that carbon out there, there's now much less steric interference. And what we see primarily in the case of our enamine is putting or having our nucleophilic carbon being even less sterically hindered than in our enolane. Okay? Subtle effect, I doubt you'll see it, but it's something worth considering. Okay? What we're getting to at this point is now just kind of talking about, well, what ifs kind of stuff. That's where we are in the semester. Okay? So within our conjugate addition reactions, our enamines give us the 1,4. Okay? That's going to be our primary situation. The nice thing about our enamines is they're relatively easy to form by just adding a secondary nitrogen, and they're also relatively easy to get rid of by adding water. Because remember, we've got an equilibrium with our nitrogens and our oxygens. They just kind of oscillate back and forth. So if I want the nitrogen in my structure to replace an oxygen, I add a lot of nitrogens. If I want my nitrogen to be replaced by oxygen, I just add a lot of oxygen. So all we really have to do to get rid of our enamine is to go through and just add water. Right? You'll typically see it added in the presence of an acid. Why? The acid acts as a catalyst. Just catalyzes the reverse reaction. Okay? Relatively simple to go through. Just because I happen to remember this from when we did this lab at ASU, the acid that typically gets added, you'll see almost always TSOH. And I've heard a couple questions about this outside of class. So what is TSOH? Not O. Toluene sulfonic acid. We talked about this back when we talked about tosylates. Okay, so let's clear everything out of there. When we talked about tosylates, I drew up the structure of our tosyl group.
We just protected acids, didn't we? Alcohols.
And we said, and I drew a squiggly line. If that squiggly line equaled chlorine, what did we have? We had an electrophilic sulfur. If our squiggly line equaled OH, we had an acidic hydrogen. Okay. So what is TSOH then? Well, if this guy is merely TS squiggle, what do we have? We have an acid. Okay. Toluene sulfonic acid is a classic organic acid. It's used instead of sulfuric acid because it has some organic character to it. So its solubility is much, much higher in organic solvents. That's our primary reason for using toluene sulfonic acid. Same general principle as sulfuric acid. It is a strong acid. It is non-nucleophilic. Okay? So don't be confused by seeing TSOH. Just because I don't think the name is on there. Yeah, I took the name off. This enamine reaction, particularly when you're doing it in the Michael type addition reaction, of course gets another name. It is referenced as the stork enamine reaction, or stork enamine addition. So if you happen to see the stork word float around, it's because some guy named, I think it's actually Gilbert Stork, happened to do a Michael addition through an enamine and stamped his name on it. Okay? So it's actually, it was probably for somebody else's graduate work. The names that you see associated with any reaction, very few of them actually did that chemistry. Okay? They were the primary investigator, and they had the graduate students go through and do that experiment. The graduate student got no fame, and the primary investigator picked up all the credit. That's the way research works.
Unless we perpetuate it.
Yep, because they're like, well, I did all that work and I didn't get my name attached to it, so I'm gonna do some more work or make somebody else do some more work and attach my name to their work. Yeah, it's awesome.
The human ego.
Yeah. Okay, so here we got a synthesis molecule. Give you some time to go through and work this out. We want to use it as the stork enamine synthesis. Okay, so this means one of your intermediates needs to be an enamine. Okay, it needs to show up in the synthesis. So go ahead and take a stab at it, see what you come up with. If you got questions, raise your hand, I'll come to you. Help. If we're trying to replicate what we've been working on, you've got this conjugate addition reaction. So let's focus on that. To do the conjugate addition, you need a double bond, an alkene, conjugated with a ketone. And it needs to be alpha beta. There's your conjugation. So there's a couple ways that we could go through and look at this. All right, we've got what I'll call my red carbonyl and what I'll call my blue carbonyl. Where's the alpha beta position relative to the blue carbonyl? That's going to be the important one, that direction. Why not go the other direction? There's no carbons over the other direction, so I don't care about doing alpha beta stuff over here. Those are technically alpha beta, but I didn't do anything over there. I'm not trying to add hydrogens to that structure. We're trying to go through a carbon-carbon bond formation. Okay, so that's one possibility for my alpha beta. The other possibility is now looking at my red structure. Where could I form my alpha beta then? Well, we have two possibilities for alpha.
But there's no beta.
There's no beta on the far right, so let's ignore that one.
So it shares the beta position with the other structure.
But don't I have that beta position up there?
Which it's not attached.
It's not attached to the other structure for the same reason we excluded the others. Okay? So which of these do we want to make the unsaturated carbonyl? Does it matter?
No.
Yeah, it does, actually.
That would be weird if the blue
So-
one, if the blue one was out
So
there.
what I'm going to do is draw them both up. In our blue situation, what we're saying for our alpha beta structure is that we've got our ketone and we've got that. There's our alpha beta unsaturated carbonyl. If it was our red one, we'd have that, but we have to watch out for our carbons. There's that carbon, and that carbon. Everybody follow all that? Okay. What would be, and so what we just drew up was our alpha beta carbonyls. What did the alpha beta carbonyl act as in our reaction? Now you got to think back to everything you've been drawing out for notes. It acted as the electrophile. So the other piece needs to be our nucleophile. So with our blue alpha beta position, what would our nucleophile need to be? It's the piece that hasn't been labeled yet. Whoops, on the blue one we're missing a carbon as well. We need that carbon. Yeah, I just didn't see it. What's the new bond that's formed from our blue alpha beta position? Where is it in the structure?
That alpha beta of the other.
Red alpha beta.
It's
red alpha.
the red alpha to the blue beta. So it's this bond in there. That's our new bond, which means the piece we're missing is where? Right there. Okay? So let's draw that piece in. Here we go. We don't want to attach it because what we're trying to show is that it's making that new bond. Sorry, I'm going to draw it a little bit higher. Whoops. Right? Everybody see that? You're saying no. What do you not see? We'll talk about why in a second. What about the red one? What would be our missing piece?
It's a
So,
phenylpropane with carbonyl.
in the red one, it's our that bond that's being made. So the piece that we're missing is this guy. So two possible situations. Everybody see those two possible situations on how we could connect that up. So the question I asked previously was, does it matter which one we do?
I do, yes, it does,
Which one is the correct one?
the right.
Why is the right one? That's kind of funny. Why is the right one correct?
The right one. Because the left,
Because if you look at the nucleophile,
It does have to do with the nucleophile. What about the nucleophile?
when
well,
it's
there's
formed,
one nucleophile, there's going to be a resonant stabilization.
You get resonant stabilization on the left one too.
yeah.
How? Because if it's tapped.
There's resonant stabilization for both those nucleophiles.
Yeah, but on the right side though, it doesn't matter either way you look.
There's
at it.
our answer.
On the left, it does.
Take a look at your nucleophile. The nucleophile is happening at the alpha position to our carbonyl, in both cases for our nucleophile. Where is the alpha position for each of those nucleophiles? There are two of them. There's an alpha there, there's an alpha there. What's the difference? In our red structure, there is no difference. In our blue structure, alpha, alpha. There is a large difference between those two. Right? So if we tried to make the nucleophile with our blue structures, our blue starting materials, we would run into the possibility of two different nucleophiles running the reaction.
And you may not.
One may be favored over the other, but that's beside the point. There's two nucleophiles. In the red, there is only one nucleophile. Which one's going to give you your higher yield? Red. Okay? Everybody see that? Make sense of the pieces. Slowly break it apart to see where those connections make and just see what you can do and build from. Okay? And if there are two options, there are two options. Draw them both out. I'm recording? Good. Just panicked. Okay? Next issue. The problem stated, so this is when we talked about synthesis. Remember, when we look at synthesis, we have to know what rules we have to follow. Okay, so we may have found our primary carbonyl pieces, but did we answer the question that was asked? The question that was asked says we need to do the stork enamine synthesis. Okay? Have we shown an enamine yet? No. The enamine was used to generate what? The nucleophile. So for our reaction to work, what we'll need to do is treat the species that we want to act as our nucleophile with something that's going to give us an enamine. So it needs to be a secondary amine. Does it matter what secondary amine we use? No. But most commonly, you'll see a cyclic secondary amine. And I don't remember what this is called. Pyrrole? Anyone remember? Pyridine? It's not pyridine. Pyridine's the 6-member group. I can't remember. It might be pyrrole.
We'll go with that.
Okay. But a secondary amine, any secondary amine will work. What that goes through and does is then generate, whoops, I need to draw that a little bit higher. Whoa, I ran out of space really fast. We'll generate our enamine, which is kind of on the screen. If you watch the video, you can see it. Now that we've got our enamine with our nucleophile, that can then go through, react, draw to the right place, react with our alpha-beta unsaturated carbonyl. Okay. We'd get the products that are shown by those guys over there. Went through and drew it up relatively well for us. To neutralize, we need to add water to go ahead and get us to our final carbonyl structure. But we also need to get rid of the enamine. How do we get rid of the enamine? Water. Right? Remember, back to our carbonyl chapters, when we're looking at nitrogen and oxygen acting as nucleophiles, they will trade each other in those kind of sp hybridized situations. So if you start with a carbonyl and you want to make an amine, you just add a lot of nitrogen. If you start with a carbonyl and you want to make an enamine, you start with a secondary nitrogen. You add a lot of secondary nitrogen. If you want to do the reverse, what do? Give it a ton of oxygen. Okay? So you are in massive equilibrium conditions with both of those circumstances. And if you want to favor one over the other, you just limit the presence of one of them. Okay? So if I want the carbonyl, I need to limit the presence of nitrogen in my reaction. Because if I have a lot of nitrogen, what happens? It goes to my imine. It goes to the nitrogen being in the structure. If I want the nitrogen structure, what do I need to limit the presence of? Water. Because water is going to take me towards the carbonyl. Okay? So you have to remember that those are in strict equilibrium, and they're going to be oscillating back and forth under all those conditions. Okay? Robinson annulations. Big fun one. This is now a massive combination of everything that we've been looking at. You've got a Michael addition followed by an aldol condensation. The classic example is shown up here, which is actually what we did in lab, almost perfectly. We've got an acidic hydrogen, which we can pull off to make our di-resonance stabilized enolate. The enolate can attack our alpha-beta unsaturated carbonyl, which then pushes us through to, I don't want to draw that structure out again, through a sequence of steps up to just our strict Michael addition result. Okay, the issue with our Michael addition is we're still under basic conditions. So this is what's neat about the aldol reaction. While we're still under basic conditions, we have to look at that structure and say, is there an acidic atom? Do I still have alpha hydrogens?
Yes.
Yeah, I got a lot of alpha hydrogens. There's an alpha, there's an alpha, there's an alpha, there's an alpha. Of those, 2 are the exact same. So I'll erase one of them. Okay, so we now have 3 possible alpha hydrogens, which could become nucleophilic. Okay, so let's go ahead and color code those. If we look at our red nucleophilic atom, where's the electrophilic location?
The far right carbon.
It's going to be this carbonyl. None of the other ones are in the correct distance to allow for a proper nucleophilic attack. So we could create the bond, which is going to look ugly, but let's just say it's going in underneath everything, straight across there, which gives us a 6-membered ring. 1, 2, 3, 4, 5, 6 atoms. 6-membered rings. Are those good?
Yes.
Yeah, so we'd say, hey, that's a possibility. There's one issue with that possibility.
Could be bridge.
That now highlighted red carbon area is a 1-carbon bridge. Bridges are not the most stable. They prevent chair rotations, chair flips, all of that fun stuff. That is not a favored situation. Okay? I'm not going to attempt to draw that, but if you want me to draw it after class, I will. Okay?
I can see it, and it's really not happening.
Yeah. If we look at our blue alpha carbon, where's the electrophilic atom?
The
Greater than 1, less than 3.
the middle, the middle carbonyl carbon.
Both of these carbonyls are valid targets.
True.
If we connect that up, either one, doesn't matter which one, how many atoms would be in a ring?
4.
Are 4 atoms a stable ring structure? Would we expect that alpha carbon to do the attack?
Nope.
Nope. So all of those are valid, but it's, do we produce something that's stable? If we can't produce something stable, then it can't occur. Okay, so that blue one doesn't get us a good result. Where's our electrophilic atom with our purple nucleophile? It's the same one as our blue ones. What happens if we do that reaction?
Get a 6-membered ring.
We get a 6-membered ring. Is it bridged? No, there is no bridging in that product. The net result is that we favor that product forming. So if we draw out our purple result, we get our 6-membered ring. Let's piece this all together. There's where our alpha was. There's our effectively aldol product. Everybody see it? Are there still alpha hydrogens?
Yes.
Yes. That alpha hydrogen can be pulled off by our base. The electrons can come in and kick out ROH.
A really bad leaving group.
As a horrible leaving group, and everybody panics about that horrible leaving group. But it's not leaving on its own. So when we looked at bad leaving groups, it was all because they were leaving on their own. This is not leaving on its own. It's being forced off. And it's being forced off into an environment that is full of bad leaving groups. Okay? So it's not a question of, oh, this is bad, it can't happen anymore. It's, oh, this is bad, but it's going into a place that's already bad. Who cares?
So bad is
Right.
happy together.
So bad is all happy together. There's another big reason that is probably a better explanation for why this reaction works. We generate an alkene, like we would in a standard elimination reaction. But this alkene is much different than a standard elimination product. Why?
It's resonance stimulus.
The answer to all questions in organic chemistry: you generate a resonance stabilized alkene. Your product went from being to wicked stable. Hey, there it is again. Okay? That more stable product is really what's driving the aldol condensation. So we can lose a really bad leaving group, and that may be extremely high energy, but the product we produce at the end of that is so much lower in energy that it accounts for the loss of a really bad leaving group. So your Robinson annulation, you string together a Michael addition with an aldol condensation, and you get this wonderful product known as the Weiland-Mischer ketone. Okay? And you just synthesized that in lab, exactly right. You did it more complicated because we did the stork enamine with a Michael addition and an aldol condensation. Okay? Synthetic patterns, just because we're almost done, and I just want to finish this. Your stork enamine synthesis and your Michael additions, if you take a look at your end result, you end up with a dicarbonyl compound. So you get 2 functional groups in your final product, and they're separated by 5 carbons. Okay? It will be a necessary result of that. And that's what this slide is attempting to show. If we look at the aldol and Claisen reactions, and for that matter, the Diekmann and there's another weird one. I don't remember the other name of it. Okay. The aldol and Claisen reactions, you end up with, again, a difunctional compound that is separated by a 1,3. So you get that chemistry again, but again, the carbon distance is now slightly different. 1,3 versus 1,5. Okay? So just slightly different chemistry coming out of each of those. So if you look at a product and you see your functional groups are separated by 3 carbons, or separated actually technically by 1 carbon, but there's 1, 2, 3 carbons in that numbering system, you probably could go through and do an aldol reaction. If they're separated by 5 carbons, you could probably go through a Michael addition type reaction. So it's a question of identifying where those functionalities are and how you could potentially string them together to get through and see that. Right? Ultimately, I think this is kind of a load of crap to go through and memorize because it's one extra thing to memorize as opposed to just looking at where your charges fall. Okay? So if it helps you, great. If it doesn't help you, focus more on where the charges are, where everything balances out. The last slide was just some synthesis question. So when we come back on Monday, I will provide the answer to the synthesis question, and then we will move into the rest of the slides, which sort of, thankfully, I didn't get to. Because because the next sequence of slides I didn't post. So, cool, worked out pretty well.